Good day. We are going to talk about DNA. Here's a picture of a DNA. A DNA is composed of the base pairs, sugar phosphate backbone, and the base pairs known as adenine paired with thymine and guanine paired with cytosine. By the way, DNA stands for deoxyribonucleic acid. Quick facts: humans share 50% of their DNA with bananas. Cells can contain 6 to 9 feet of DNA. If all the DNA in your body was put end to end, it would reach to the sun and back over 600 times. Ganyan ka haba yung ating DNA. DNA in all humans is 99.9% identical. It is about 1/10 of 1% that makes us all unique, or about 3 million nucleotide difference. DNA can store 25 gigabytes of information per inch and is the most efficient storage system known to human. So humans are better than computers. In an average meal, you can eat approximately 55 million cells, or between 63,000 to 93,000 miles of DNA. And it would take a person typing 60 words per minute, 8 hours a day, around 50 years to type the human genome. So what do we know? DNA is composed of units called nucleotides. Now, a nucleotide is composed of these three molecules. Okay, we have the phosphate, pentose sugar, and nitrogenous base or bases. Now, in every nucleotide, there's one particular base that would match to the complementary base. What is pentose sugar? Pentose sugar is one of the backbone of the DNA, which is known as the deoxyribose sugar. Deoxyribose, or more precisely 2-deoxyribose, indicates that it is a deoxy sugar, meaning that it is derived from the sugar ribose by loss of an oxygen atom. Okay? Now, deoxyribose is most notable for its presence in DNA. 1-phosphate, the nitrogenous base. So, the nitrogenous bases are categorized into two, namely purine or pyrimidine. This you have there two different bases. Okay, so this one and this one. Later on, we'll discuss ano nga ba yung pyrimidine and purine, nitrogenous base. So you have there the pentose sugar, also known as the deoxyribose sugar, and the phosphate molecule. So you have there the adenine molecule. Okay, so it is paired with deoxyribose and a phosphate molecule, and this whole thing is known as the nucleotide. Okay, again, a nucleotide is composed of, you have there the phosphate, the carbon, and the pentose sugar. However, you have in this side, known as the nucleoside, only the adenine base or the nitrogenous bases, nucleoside and nucleotide base. Okay, so you have here an illustration about the DNA. So on the left, you have here the phosphate and sugar molecules bonded with each other. And on the right, the same, but in a reverse manner. Okay? So at the center, you have the base pairs. So A pairs with T and G pairs with C, and so on. Okay? DNA is composed of two complementary strands of nucleotides joined by hydrogen bands. So at the center, you have here the hydrogen bands, okay, of the two nitrogenous base pairs. And by the way, as you can see, you have here two bands between adenine and thymine, and you have three bands between guanine and cytosine. So always remember that the number of bands depend upon the uh, type of pairs. Okay, so if A pairs with D or this pair, it will follow that there will be two hydrogen bands. However, if the nitrogenous base guanine is paired with cytosine, it will create three hydrogen bands. So this is try to remember this base pairing. Okay, so adenine will always be paired with thymine in the case of DNA. Okay, so AT or TA, and they are joined by two hydrogen bands. On the other hand, cytosine will always be paired with guanine, CG or GC, vice versa, and they are joined by three hydrogen bonds. So DNA twists into a double helix. So this is the twisted version of the DNA, okay? And this is an illustration of the DNA. So always remember that A is paired with D and C is always paired with G.
Base pairing. Now, what is base pairing? Base pairing normally is the pairing of two nitrogenous bases like adenine and thymine, which creates hydrogen bonds. In the case of cytosine and guanine, you have there at the center, it creates three hydrogen bonds, the red ones. And in adenine and thymine, it creates two hydrogen bonds. So that is base pairing. Now, where can we locate the DNA? All cells do have DNA, and it is located inside the cell's nucleus. So, if we unwrap the chromosome, you have there the chromosome inside the nucleus. If we unwrap it, it will reveal the twisted DNA of the cell. So that's our DNA if we unwrap our chromosome.
The DNA functions. Number one, DNA directs the machinery of a cell to make specific proteins, and therefore DNA indirectly controls all of the functioning of all living things. Now, you have to take note, class, that all living things require proteins for its normal functions. Since DNA directs the machinery for the synthesis of specific proteins, therefore DNA controls indirectly all the functions of all living things. Now, all cells do have DNA, and DNA is situated on its nucleus. Now, as you can see, you have there the DNA, and it's or transcribed into an RNA.
That RNA goes out of the cell's nucleus and carries the information in a form of mRNA, and it attaches with the ribosome, which then initiates the production of amino acid polymers. So this amino acid polymer subsequently becomes our protein chain.
What is a stem cell? Now, a single cell that can modify itself replicate and modify itself into different cell types is called a stem cell. Perhaps that cell becomes your skin, your blood, your neurons, and so many other types of cells. Now, the second function of the DNA is to store the hereditary information of an individual. Okay? So, for instance, that single stem cell differentiates itself to become a red blood cell, for instance. Therefore, there's a specific information or command from the DNA inside that stem cell, making it or resulting for that cellular differentiation of that particular stem cell. Third function of the DNA is that it allows new characteristics and abilities to appear which may help an individual to survive and reproduce. So that um, alteration or modification of new characteristics and abilities is what we call evolution. Okay? So DNA has the ability to mutate or change. So, for instance, that particular cell produces a specific kind of protein for a particular function of that cell or cell type. There's some instances that the DNA or the message itself will be modified and cause the mutation of that particular cell. Okay, so for instance, you have there the original and correct copy. Okay, so if the nitrogenous base green, for example, pairs with that violet, in the mutated copy, you'll have that original green. From original green, the mutated copy will become red. So there's a single that single alteration modifies the whole function of that particular cell. Okay? That's what we call mutation. Function number 4. Self-replication. DNA has the ability to make copies of itself. Yes, DNA replicates itself. So this is an example of it. You have the four nitrogenous bases. You have the first pair C and G, and you have the second pair A and T. Okay, so we have the so-called DNA replication process wherein a single DNA, as you can see on the right illustration or picture, you have there a single DNA unzips itself to allow the new strand, okay, to be created and allow or results to DNA replication. Okay. Now, what is DNA replication? DNA replication is called semi-conservative. So, semi-conservative means that the replication process starts with the original strand of the DNA and it will remain intact and act as templates for the synthesis of duplicate strands of DNA.
One strand of the new daughter DNA is from the old parent DNA molecule.
That's called semi-conservative replication. Now, one copy of a DNA molecule will split apart to make two complete copies of itself. Each new DNA molecule is made up of half of the old molecule and half of a new molecule. Meaning, yung kalahati ng bagong strands ng DNA, meron siyang one half of it, merong old molecule strand, and half of it is new molecule. Okay? For instance is, itong kulay blue, this one's the new one. Okay? And the red strand is the old one. Steps to DNA replication. So, nandito na yung sa activity ninyo. That's activity 2A. Okay? Now, the first step for DNA replication is unzipping. So, paano nga ba nagsisimula yung DNA replication? DNA replication starts with the unzipping process of the DNA strand. Okay? So, the DNA molecule unzips as hydrogen bands between the base pairs are broken. So, katulad ng nasa picture na yan. So, the DNA unzips and break the hydrogen bonds between the two um, pairs of nitrogenous bases. The enzyme helicase then causes this unzipping to occur. So, the next step is complementary base pairing. So, simply, yung mga complementary nucleotides lalapit doon sa mga old strands ng DNA and it matches with a nitrogenous base. Okay? And bond with that particular nitrogenous base. Okay? So, third, form new sugar phosphate backbone. So, the nucleotides join as the sugars and phosphates bond to form a new backbone. This process occurs due to the enzyme DNA polymerase, which also checks for mistakes as it goes.
So you have different shapes in that illustration. The yellow circles represent the phosphates. The red pentagonal shape represents the deoxyribose sugar known as pentose. And you have the represent the shapes represented with or labeled A, C, T, G. Those are the nitrogenous bases. So every sugar phosphate backbone carries with it one nitrogenous base, which matches with the other strand. It somehow complements with the other strand, thus creating the double-stranded DNA.
The process continues along the primary chain until we have two identical strands of DNA molecules. This process assumes that there's no single error made. Okay, so yan yung product niya, dalawang daughter DNA molecules.
Regarding sa activity ninyo, you are going to somehow recreate the complementary base pairing using the template. But first, you are going to cut it out, yung mga shapes na nasa template ninyo, you are going to color it, and you will do complementary base pairing by matching the nitrogenous bases, okay? Adenine with matches with thymine and guanine matches with or pairs with cytosine. But always remember that if you are going to do DNA transcription meaning you're going to create an RNA, there is no, remember that there's no thymine in RNA. Instead, you are going to replace thymine with uracil. So, adenine pairs with uracil in RNA transcription. That's it and have a good day.